Epigenetics is the study of how environmental circumstances and life experiences can turn genes on or off without changing the actual DNA sequence. So, why exactly is this important? It's crucial because it teaches us about how our lifestyle choices, such as our diet, sleep quality, or experiences, can switch certain genes on or off. And these are genes that can influence health, disease risk, development, and aging. This offers us a new path for personalized medicine and understanding inheritance beyond just the DNA sequence. Through our life events, we can modify our genetic destiny. How does this work? So we all have cells, and within those cells we have DNA. But before the DNA condenses into chromosomes, there's a level of organization it needs to hit. The DNA wraps around a core of 8 histone proteins, and this forms a packaging unit called the nucleosome, resembling the beads on a string. Now, epigenetics doesn't change the actual sequence of DNA, but it works by adding chemical tags to DNA and its associated proteins, which can turn genes, aka sections of DNA that contain instructions for protein synthesis, on or off. DNA methylation is one way that our genes can be turned off or on. It works by tagging DNA with methyl groups. This silences the gene and prevents it from synthesizing protein. Now, histone acylation is also a way that DNA can be modified, and this occurs when an acetyl group is added or tagged onto a histone protein that the DNA is wrapped around. This can change how tightly or loosely the DNA is wound around them, and since these tags typically neutralize the positive charge of the histone tails, the DNA will uncoil. The uncoiled DNA makes genes more accessible and can increase gene expression. Now, how exactly do our diets affect this though? So, various studies have found strong evidence that supports the belief that our diets can greatly influence the processes that are directly responsible for the on and off switches of these genes. So you really are what you eat. That really applies in this situation. And foods that are described as being rich in methyl groups enhance the process of DNA methylation, which can turn a gene off. Nutrients such as folate, B12, B6, and more are rich in these methyl groups. Dark leafy vegetables, beets, eggs, liver, legumes, nuts, berries, and whole grains also provide adequate amounts of these methyl groups. Now, foods that aid in histone acylation are foods again such as green leafy vegetables, berries, grapes, green tea, turmeric, apples, onions, and more. These foods again unwind the DNA from around the histone proteins, making the genes more accessible. Now, a great example of this is actually green tea. As we know, green tea is a very powerful antioxidant that holds the compound ECGC. This influences histone acylation by inhibiting deacetylase, leading to loosely compacted chromatin and accessible genes. Green tea acts as an epigenetic modulator by rewinding the abnormally silenced genes that are often seen in various diseases. A lot of these diseases are cancer, diabetes, or even mental health disorders. That's all I have for today. I hope you learned a lot, and I do want to point out that all the images I use are from Creative Commons. Thank you.